Slides again. Okay, yeah, awesome. I'm really honored to be here. It's really fun, I think, to be able to give a talk to a different set of audience that I'm usually used to. So really looking forward to this and see how you guys respond and engage with the kind of work that we have been thinking about. So as Yona said, I'm a graduate student in Terry Hua's lab, though not for much longer. And today I'll tell you about some of my doctoral work, which has had different thrusts, but the main theme that's been consistent is thinking about how bacterial growth happen on heterogeneous substrates and on particles in particular. Chitin has been the example that we've used and studied to the most detail, but we've also thought about all sorts of heterogeneous substrates as well. And I guess to jump right in, one of the main differences that happens when one starts thinking about structured substrates and things that have spatial structure is that now all of a sudden the idea of localization becomes important. And because cells have, a finite size, they cannot be like both here and there. This is the statement that I like to say: there is no quantum cell. A cell has to really localize itself if it wants to perfect function. There tends to be this thinking in the ecological field that trade-offs between being able to colonize, and so being able to grow on your current substrate, as well as being able to disperse in order to explore what else is out there. And this can be apparent in a few of the vignettes that I'll tell you about in a little bit. So one way one can think about this is that, for bacteria that grow in liquid environments with multiple substrates, they can sample, pick and choose whichever substrate they prefer to grow on, and then adjust their regulatory strategies to grow on, I don't know, glucose versus glycerol. However, if the substrates are trapped physically and have, like physical structure, this kind of sampling cannot happen. And so there is really a commitment, a regulatory commitment that has to be made by the bacterial cell to grow on a specific substrate versus another. In certain environments, cells must survive long enough and start exploring their environment before they encounter the next environment. And this is just because resources are finite in most environments. And so in the example of chitin particles, for example, a cell might want to explore the possibility of finding another particle that it can grow on before that particle is degraded and before the cell finds itself starving. In the ocean, as you guys are familiar with, gravity plays a role, and so things are sinking down. And because of sinking, one can imagine that a certain bacterial population might both want to maintain the population along the water column, as well as avoid some of these, like in hospital labs and the ocean. And so these are just a few vignettes that I think can help us think about the existence and the possibility of the existence between these colonization and dispersal trade-off. And I guess a common theme is that there is some bet hedging that has to be made, although this is not the framework that we'll be using for discussing these things. But you have a substrate currently, but then there's also all of these potentially detrimental things are happening to you in the future. And so one has to make sure to maintain the population across timescales that are longer than those on which growth and replication happens for bacterial cells. And so for the case of chitin particles, we decided to write down a pretty detailed microscopic model to try to understand how these things work. So I want you to imagine a chitin particle that lives, in an ocean. Usually the size of the chitin particles that we're thinking about is about 100 micrometers. And so we have these small cells that are attached to these particles. And because of the differential in size, we can simplify this problem into a one-dimensional problem where we only consider the height, or I guess the how far away a bacterial cell might be from the surface of the chitin particle. I'm presenting a 1D model, but we've been able to solve the full 3-dimensional structure with spherical symmetry. So we have these cells that are coating the chitin particle, and this microscopic model, what we wanted to do was just to kind of make an inventory of all the possible things. So propose a detailed model that doesn't have to invoke anything too magical, other than just processes that we know to happen in these kinds of cultures. So a cell that might be on the particle can have a detachment rate, producing a planktonic cell. These planktonic cells can reattach back to the chitin particles. Now, these cells, depending on how fast they're growing, are going to be making chitinases, which are the enzymes that we know to degrade chitin. These chitinases are producing nutrients with a certain rate. And then to kind of close back the loop, the nutrients are consumed by these bacterial cells, which aids in their replication and therefore in the creation of more enzymes, and this aids for growth to proceed. And so we can write down a ton of equations that I'm just going to flash, happy to discuss the details of some of these, but I also want to draw attention to some important details of how we're formulating the equation. So we have this nutrient profile that includes both diffusion and uptake by the cells, a cell profile that includes diffusion, cell growth, as well as cell chemotaxis. I want to draw your attention to the fact that the chemoattractant in our case is also the nutrient, and we know this to be true for, I guess, the bacterial strains that we are studying. And the chemotaxis term has an interesting little modification or addition that we know from previous work on E. coli cells to be true, where there is a cutoff to the absolute concentration of nutrients that the cells can sense. So this is represented by this K chi here. And on the surface, we have these surface attached cells that can both replicate and then are subject to exchange dynamics, so they can attach and detach. And we have surface enzymes that are being produced with a certain rate, and this rate depends on the replication rate of the specific cell. So we're thinking of these enzymes are just, another set of proteins that the cell can make, just like ribosomes. As the cell is dividing and replicating, it is also a part of its regulatory strategy is also to make enzymes and chitinases. And, because this is a spatiotemporal model, we have no flux boundary conditions on both of the boundaries, as well as a set of initial conditions. So, you can write something like this. There is nothing too magical about the recipe that we're invoking here, except that this becomes complicated very quickly. There's tons of parameters. And in particular, these are all single cell parameters. It would be nice to know what the single cell attachment detachment rates were, the single cell production flux of chitinases is, and all of that. And unfortunately, this is very difficult to do. We've tried doing this a little bit, and I'm not sure this video is going to play. Oh, it does, okay. So you can take videos of bacteria detaching from chitin particles and perhaps track the cells and all of that. That's doable, quite difficult, and we also don't have the right microscopy set up for this kind of work. So that's one approach that one could take, like really try to characterize all of the single cell parameters. The other thing, though, is that even if you're able to access this kind of information, it might not be the most useful, because what one cares about, perhaps on a little bit more of a macroscopic scale, is something about population flux and population exchanges rather than single cell dynamics. We can do a little bit more microscopy and look at these particles and look at how they're colonized by some of these bacterial cells. And the geometry of the particles is quite complicated. So, even writing down a simple model might not be the best thing to do in that case. And then I want to note, this will become relevant in a second, but even at high densities, for example, in this particle here, you see that it's not fully colonized. So there's, like very specific things happening on the surface of these chitin particles. And let's say one had access to all of these like single cell population dynamics, it is very difficult to measure things like nutrient concentrations and enzyme concentrations. And so because of that, we kind of decided that this microscopic approach was perhaps not the best way of approaching this problem, especially if we're thinking about it from a more general macroscopic sense. And so instead, we suggested and we developed a simple experimental setup that helps us assess all of these population level dynamics. And so our experimental setup is very simple. We have we're inoculating these Vibrio cells. They're a nice natural isolate from a chitin degrading community. So they are chitin degraders. And if you look at their genomes, they have 3 different chitinases and all of that. We also know that they secrete their chitinases extracellularly. This is an important feature of the specific bacterial strain that we're using. And we're incubating them, we're feeding them chitin as the sole source of nutrients. So there is no other source of carbon or nitrogen in the culture. chitin is thought of as very hard to degrade. And so in order to grow, these cells better express their set of chitinases and start the degradation process. That's the only way for them to grow and accumulate biomass. When we set up this experiment, we didn't really have set expectations about what this should look like and what our data should look like. So typically, when one thinks of bacterial growth on dissolved substrates, typical growth curves look something like this, where you have in semi-log plot exponential growth, so a phase of linear growth in the semi-log plot, and then the culture saturates. But we in this case, when nutrients are not dissolved, they're trapped in this physical form. We weren't sure exactly what kinds of growth curves we would obtain and if we would be able to extract relevant information from there. And so there are all these questions about where is growth going to be localized? Is it going to be on the surface of the particles, or is it planktonic in the bulk? And what are the timescales of this degradation? In the literature, people think of chitin as a somewhat difficult task, or at least harder than just assimilating dissolved carbons and dissolved sugars. And so for this specific strains, what are the timescales? What are the length scales? So let's just dive in. One thing that we can do after we jump over some experimental hoops that have to do with the fact that chitin itself has an optical density because it's a colloid that's suspended in solution. So what we can do is centrifuge a lot of our samples at low speed, decant them, and then measure what we're going to define as the planktonic OD. So this is the OD of the cells that are in the bulk. And so when we do these experiments, what we see is these kinds of growth curves. On the y-axis, I'm plotting planktonic OD. On the x-axis is time. And what we can do is take some of these planktonic cells and then start a new fresh chitin culture, so incubate them with fresh chitin particles. And what we see is that we can recover the same exponential growth rate. So we have a population that seems to be, at least in the planktonic phase, growing planktonically. The timescale of this population growth is about 12 hours of doubling time. And so we can write down, this equation where NB, which is the total number of planktonic cells, is increasing exponentially at this rate, lambda, that we've measured. But the planktonic side of the population only tells us so much. It tells us what part of the subpopulation is doing, and we know that we're feeding these particles chitin, so most of the interesting biological things are probably happening on the surface of these particles. And so in order to access this kind of information, we used RNA measurements, like one way of seeing how much of the biomass is on particles. We were able to measure both RNA amounts directly, but experiments that I'm going to show you relied on measuring the incorporation of radioactive phosphate into the cells and into the biomass, and then tracking that both in the planktonic and in the surface attached populations. So the kinds of experiments that we can do are subtractive, just because it turns out that the biomass on the surface of the particle is not enough. So we can take samples that contain both planktonic cells and surface attached cells, as well as samples that only contain planktonic cells. And these are the things that I'm plotting in here. The black dots are the total population, and the blue dots are the planktonic population. See that there's consistently a difference between the two. And if we take the ratio of these blue points to the total points, it tells us something, I'm skipping ahead. It tells us something about the fraction of the total population that it's in the that is in the planktonic state. So we're in the situation where we think both of these subpopulations are increasing exponentially. They're increasing at the same rate. They're just exchanging. And that, surprisingly, even though the only source of nutrients in our culture is on the chitin particles, the majority of the population is planktonic. And in fact, 3/4 of the population was planktonic. And so this was a little bit puzzling to us. How is it that we're feeding these bacteria, so much chitin? They have the ability to degrade chitin, but somehow they're choosing, to detach from these chitin particles and to not localize themselves to them. So we can go back to our detailed model and then start thinking about this in perhaps a way that's a little bit different. So one thing that we can do is to say, okay, let us ignore for now the spatial component, because we observe exponential growth of the cell profile. We can just ignore all of the spatial component and call this entire population NB. So this is the NB that we're measuring. It's the total population of planktonic cells. Similarly, we can simplify a lot of these other fields. So the surface cells are just a number or surface density of cells, as well as the enzymes. And we can then write down and simplify the nutrient equation as nutrients being drawn out because of the replication of both of these subpopulations, but the nutrients are generated by the enzymes, and there's potentially some diffusive loss, some parts of the nutrients that might not be re-assimilated into biomass. And so what we can do then is, clean up this kind of model a little bit, and then we can start writing down similar equations, but that have slightly different meaning, right? So notice I was writing kappas for the rates earlier. These were the single cell rates. But now in this case, I'm thinking really about population level fluxes. And so even though it looks like I'm writing down the same, I'm putting together, the same picture, these are actually populations of cells and population exchange dynamics. So we have these surface attached cells that are replicating and then exchanging with the same attachment detachment dynamics, planktonic cells doing the same as well. They are producing enzymes, and then these enzymes are producing nutrients, are feeding, both of the populations and giving a replication rate to each one of these subpopulations. So with a model like this is like a population level model, and this is something that's a little bit more accessible to us experimentally. We can try to measure some and constrain our model by measuring some of these rates, these population level rates. So I'll just give you a flavor of some of the measurements that we're able to do to constrain this kind of model. So we can measure, for example, the catalytic rate of the chitinases, this rate Ke. The way we do this is that we can isolate parts of our culture that are in exponential growth and then prevent the uptake of glycnac by some of the planktonic population. And so for different ODs, we can look at how fast glycnac is accumulating in the media because now it's not being uptaken. And so this gives us a sense of how fast these enzymes are working. And so we can measure exactly what this number Ke is. What we can also do, for example, is to ask ourselves, are these cells, these two subpopulations of cells, replicating at the same rate, and are they replicating at all? Are the planktonic cells replicating in the sense that do they have access to nutrients? And so one way in which we can assess that, we've tried multiple ways, but I'll show you one, is to say, okay, let's isolate all of the particles inside the dialysis bag that would allow for glycnac to exchange between the planktonic cells and the cells that are inside of the bag, so that are attached to the particles. And let's ask whether the planktonic population outside of the bag is going to be able to replicate and grow. And so when we do this, we find that outside of the population, there was no change in optical density, meaning that these cells were not able to access any of the nutrients generated and not able to grow, whereas inside the growth was able to resume with the same exponential growth rate. So this really indicates that this planktonic population that is away from the particle and the sources of food is probably just hanging out there, starving and not having access to nutrients. We can also measure other things. So this allows us to kind of cross out these terms, such as the replication rate, RB, from our equations. We can also measure directly the detachment rate. So we can start and constrain this number. I'm not showing the experiments here. And conversely, what we find is that when we try to measure the attachment rate, it is actually quite small. And so these are terms that we can ignore. And so what ends up emerging from this kind of analysis is that we have a situation like this, where since these cells do not have access to nutrients, they're not replicating. And so we think that there is minimal diffusive flux away from the surface of the chitin particles, meaning that most of the nutrients are taken up by these surface attached cells to both replicate, produce biomass, and produce their enzymes, and that there is this kind of unidirectional flux of cells that are attached on the surface of the particles into planktonic cells. And so kind of the reason why we're seeing this exponential increase of the planktonic fraction is that these cells on the surface of the particles are replicating and fueling the growth of the entire population. And if you remember the statement that I was talking about earlier, something about colonization and dispersal, if we look at our second equation, we can extract, since we know, that the population is increasing exponentially, we can extract a constraint between this population level growth rate and these rates that are happening on the surface of the particle. And so really what this statement means is that lambda suffers from having a high detachment rate, KD. And if we actually put numbers to this, given what we've measured that lambda is about 12-hour doublings. We've measured a pretty high detachment rate KD, which is consistent with this picture that cells are being really pushed away from the surface of the particles. This gives us a prediction on how fast the replication rate on the surface of the particles has to be. And this is quite surprising. We started out by thinking that this entire culture was growing on a timescale doubling every 12 hours. But if you really think about the replication rate of the cells, it's actually quite fast. This corresponds to a doubling time of about 3 hours. So these cells on the surface of the particle, they're growing faster than what one might imagine. And because there is such a high detachment rate, the apparent exponential growth rate of the population seems to be lower than what it might be without this detachment rate. So this is kind of the population description that constrains lambda, this population growth rate. However, if you look at our fourth equation, it says something quite profound, which is that the exponential growth rate, this lambda, has to be proportional to these enzyme characteristics. So to Ki, which is the allocation of cells to making chitinases, you can think of this as just the amount of chitinases produced, multiplied by the rate, the catalytic rate of GLICNAC production. So how many enzymes versus how fast they're making GLICNAC, right? And this is a little bit more of a molecular description of what this population growth rate is. But this molecular description is a little bit surprising because what it tells us is that lambda does not depend on these population exchange dynamics. And in particular, it does not depend on KD. So if you take seriously this left-hand side of the equation, you could get away with as fast of a KD as you want. So you can imagine a population that detaches infinitely, but that would still be able to grow. Because if the bottleneck to growth is just generating enzymes that generate nutrients, then it is intuitive that that's what regulates and what sets the growth rate of the entire population. And this puzzled us for a little bit. This was a bit of a surprising kind of take-home message from this kind of simplified analysis that just accounts for exchange dynamics of cells. And so perhaps we understand it mathematically, and so perhaps to build a little bit of intuition as to why that might be the case. Let me show you a picture. So we imagine that we have, some a chitin particle with some surface attached cells that are secreting enzymes that are stuck on these chitin particles. After some time, they're going to double, so replicate and double. So both the enzyme and the cell numbers will double. And let's consider two situations. One in which the detachment rate is low. So some of these cells are going to detach, and the remaining cells are going to be able to replicate with a certain rate, RS. On the other hand, we can imagine that the detachment rate is way faster. So a lot more of these cells are going to detach. And so we're left with surface attached cells that now are taking advantage of a larger amount of enzymes. And so the nutrient concentration per cell is now higher. And so this indicates that there is a trade-off between the replication rate on the surface of the particles and their detachment rate. So these two things are co-varying in a way. So if we look back at our other equation, we are able to kind of solve this dilemma, this colonization dispersal dilemma, by saying that RS adjusts to the nutrient concentration through adjusting to KD. And so these terms balance out such that a higher detachment rate also results in a higher replication rate, maintaining the overall population growth rate, and that the population growth rate is only set by enzyme dynamics, right? So the more enzymes you have, the faster you're growing. The faster your enzymes are, the faster the population is growing. But the individual, population exchange dynamics do not play as important of a role in this situation. And I want to emphasize here that this trade-off works because the cells have figured out a kind of clever trick of secreting their enzymes extracellular, or they're depositing their enzymes, and in this way they're decoupling their localization from where the nutrients are being generated. So as the cells depart and detach, they can still the enzymes that they've secreted previously can still generate nutrients on the surface of the particles. So now this, begs the experimental validation question. Can we see this? Can we change the detachment rate and then see that this lambda, this overall population growth rate is conserved? And unfortunately, this is not something we can do because we do not have good control over the genetics of our population. However, we can go back to our detailed model and then do these experiments in silico. And by that, I just mean changing parameters in our model and then seeing how lambda responds to this microscopic change in the parameters. And in fact, when we do this, we can change, for example, phi E and Ke, which are the two things. Phi E is the same thing as Ke previously, so just the allocation towards making enzymes. We can see that there's this proportionality between lambda, the population growth rate, and enzyme properties. However, with cell properties, things do not vary in the same way. And in particular, as we increase KD, the growth rate is not changing so much. The reason why we have a slight decrease from our expectation here is because we have a non-zero attachment rate in these microscopic models. And we can also ask, for example, the question, how does this lambda depend on the chemotactic bias? So if the cells can chemotax more towards the particle, how is that going to affect the overall growth rate of the population? And it turns out that it does not very much. And so this model really allows us to, start thinking about how all of these microscopic parameters might affect the macroscopic parameters that we care about and that we can actually observe experimentally. So it is a little bit reassuring to see that, these predictions at the population level that we did after a ton of simplifications, removing all of space and all of that, are borne out by the microscopic model. We can recapitulate the population dynamics I was showing you before. So what I'm plotting here is the total population levels as a function of time. And so given these empirical parameters that have allowed us to constrain our model, we can recapitulate the growth rate as well as the ratio of planktonic cells to surface attached cells. So the planktonic cells are in green and the surface attached cells are in red. And we also have some prediction about how the enzyme number is increasing as a function of time. However, now that we have this model, it's quite easy, perhaps easier than experimentally, to go and change a bunch of things in our model. In particular, start thinking about the initial conditions. And so one thing we can do is to say, okay, let's change initial conditions. Namely, let's start with the lower level of surface attached cells. So note that the scales here are quite different. And let's see whether we can obtain exponential growth. And it turns out that the existence of a steady state solution is not always guaranteed. The system does not always go to exponential growth. And because, we can do this on a computer, let's be a little bit more systematic about this and examine the initial conditions. So I'm going to show you a few graphs, a few two-dimensional graphs that look like this. So I'm taking, my four-dimensional system that has nutrients, planktonic cells, surface attached cells, and surface attached enzymes, and then changing, keeping all of the parameters fixed. And then the only thing I'm changing is the initial condition. So the initial number of surface attached cells and enzyme attached cells. And enzymes are attached to particles, sorry. And so if we start, for example, from a point like this with a lot of enzymes and not that, with a lot of cells and not that many enzymes, typical trajectories will look like this. The number of cells attached to the particles decreases because we have this high detachment rate. And at some point it cannot do better, and so it crashes. On the other hand, we can start with another set of initial conditions where we have the same number of initial cells, however, a little bit more of enzymes, and all of a sudden growth can resume. And we can do this systematically and tile this entire space. And the phase diagram, if you will, looks like this. But this is a little bit, too complicated. So we thought maybe there can be a simplified toy model that can allow us to recapitulate this kind of behavior. And so we can just simplify our very detailed four-dimensional model that was accounting for, both nutrients and cells that are planktonic and cells on the surface of the particles into a two-dimensional system where we're only keeping track of, which is a proxy for NS, the number of cells on the particles, and of the enzymes on the particles. So we're really throwing away all of the planktonic part of the system because, we think that the planktonic cells are not replicating, not generating that many enzymes, they're not that important for the growth, and really focusing on the particles. And the linearity that emerges from the system is that the enzyme concentration in this way depends on the numbers on the surface of particles. And this just means that, as you have enzymes, you're generating more nutrients and therefore able to replicate faster. So there's this dependence in the structure field that can relate how fast the cell is able to replicate and to the enzymes. So there's this couple. And one model, we can solve it exactly, and we find that quantitatively we can recapitulate both existing fixed shapes and quantitative the location of the fixed point from our mic model. So it looks like a pretty good job at quantitative recapitulating in features that we were observing health model. And so we can do is thinking about separatrix, and we start to develop an intuition for has this form. So for example, look here, your densities, a lot of these trajectories are going to follow a power law, so going to be decreasing. Intuition there is that as a population, you better be detaching from the, you have to overcome this very high detachment. So the rate to generate enzyme will be indicated by the slow diagonal and detach. And as you cross over matrix, maybe generated to allow growth to happen. These are examples that show this. On the other hand, enzyme densities, small number of cells is going to, the initial number of cells going to be enough to lead you to, even though attaching point is going to generating nutrients. So this other here of the base diagram. This a little bit unrealistic. What we're saying is that, okay, these cells are arriving on a particle, and at point of our cells and enzymes, which is not very realistic because, there's only a finite amount of it can be produced by a single cell. And so let's constrain these initials a little bit. In particular, little cells arriving to specific particles and enzymes and they have a certain amount alpha of enzymes within there. And so one way we can think of the diagram is I was showing you just through the separatrix, there is extinction phase, growth phase. But let's scan this, right? So this just means that, there is only, you can only as many enzymes as you have cells, right? And depending on more of these cells, we can look at the time trajectories, but importantly, what it has to do is to reduce this two-dimensional phase transition into a 1D phase transition that only depends on the number of cells, right? So we can project this entire phase to these lines, and then above a certain criticity, we would get growth, and below a certain critical cell, the stem. So dimension. One might, okay, well, this, the are realistic. Another set of initial conditions we can consider to introduce a little bit more to how fast it takes for these cells to dump the particles, right? We can embed that in initial conditions. By that, I just mean that there is some parameter beta that is going to determine how fast enzymes at the surface of the particles, and we can do similar kind of analysis and look at how this the critical kind of density, cell density on the particle to play out how chemotaxis might affect birds, etc. And one interesting thing is that chemotaxis does not affect the steady state, affect the initial conditions. We have this gradient of imbalance of field. And so if we have, like concentrations that have specific gradients, the cells can navigate back towards them. Finally, perhaps the most realistic set of initial conditions is our chitin particle is in the path of low level density of cells, and we can turn on the dynamics and have attachment, detachment rates, as well as the production of enzymes, etc. And then look at how this critical phase that this critical density depends on parameters such as the chemotaxis bias, as well as the cells are able to produce their enzymes, and we see that there. Just to summarize this bit a little bit, what we've uncovered is that in thinking about the initial dynamics, so we were able to characterize the steady state growth in our experimental work as well as for the modeling. But then thinking about the initial transients, the initial dynamics, we have uncovered that there is two parameters that really matter for the possibility of growth or extinction on the surface on chitin particles. This might be generally true for other kinds of solid substrates, and that this two-dimensional initial condition phase diagram determines whether the degradation of the particle can happen to completion or not. And there's just a few ingredients that are necessary to produce this Allee effect, which I've shown you in the 2D toy model. And to summarize them, it's basically that we need this dependence of the nutrient structure. So the growth rate of single cells have to depend on the density of cells and therefore on the density of enzymes. And we have a high detachment rate, so the population has to surmount this high detachment rate, but to generate enzymes fast enough. And the other interesting feature that generates this kind of, from a very simple model that generates interesting dynamics is that we have enzymes that have a finite lifetime. And so I think, okay, perhaps, yeah, quite on time. So I just wanted to sum up a few of the things that we've discussed today. So when one starts thinking about the degradation of things that are localized, that have, spatial structure, there is this trade-off between growth and exploration, so the cells can either consume their local nutrient source or detach to explore other nutrient sources. And what we've shown experimentally, I guess, in our system is that we have, a strain that prefers to detach and therefore prefers to explore. However, in order to maintain the possibility of growing on its current source, it's also secreting these extracellular enzymes that allow for growth to continue. So this nice mechanistic feedback between the replication rate on the surface of the particle and the detachment rate is what allows for the population to grow. And that really just leads us to think about, what local parameters really affect the steady state growth and the population level growth rate. And what it turns out to be is that really enzyme dynamics are what constrain the rates at which chitin degradation can happen. On the other hand, there is potentially an Allee effect. So, this exponential growth does not always happen. It depends on the specific initial conditions, whether the cells are able to store enough carbon to generate some chitinases and things like that. And so these initial dynamics really have to fight off the high detachment rate that we're observing. And so because of that, they create this interesting positive density dependence in the system. So the more initial number of cells you have, the easier it will be for growth to occur. And yeah, so with that, I'd like to thank my advisor, Terry, who's been super helpful throughout this whole project, as well as some collaborators. A lot of the experimental work has been described in this paper that came out last year in Nature Microbiology. And we are working on finalizing some of the theoretical work regarding the Allee effect in the publication that will be happening soon. I would also like to thank people in this prime collaboration that we're part of, Jonas for inviting me, and then all of these other people for very, very useful discussions over the years. And with that, I'm happy to take questions. Marco, I think, yes. You can self-moderate, I guess.
Nice catch. So thanks for the talk. It was really nice. And I have I had a bunch of questions throughout the talk, but I forgot many of them. But one of the things that I thought you were going to talk about, and in the end it didn't come out, is this observation at the beginning that you have clusters on the particles, right? So do you want to elaborate a little bit on that? Because that's not something that really is being addressed in the model at the moment.
Yeah, absolutely. I mean, in our models, we assume this uniform density, uniform coverage of the cells on the chitin particle, and clearly that's not true. One thing that we're thinking about, though, is that the size of these clusters can allow us to kind of simplify and to think about local areas of the chitin particles where the distribution of cells is homogeneous. But if you look at the pictures, that's clearly not the case. I think in our case, we just don't have the experimental possibility of really in detail looking at some of, doing like careful statistical analysis of what the shapes and sizes of these clusters are. So this is not something we have addressed directly. But you are right that it might create interesting dynamics. And namely, if you look at if you try to solve some of the models that we have and think about, are there any feedbacks, for example, that help localize the cells to where the enzymes are? And if when we do agent-based simulations, we see that. And when we think about our models, we see that there might be feedbacks there. So what I mean is that, you can have a certain cell that deposits an enzyme. This enzyme is now generating nutrients. Cells will attract towards these nutrients, and therefore the colonies will grow locally. And so that helps kind of localize the entire dynamics to certain regions of the chitin particles. But this is not something we have looked at in detail. Yeah. The other interesting thing is that depending on, the specific nature of the substrate, in the pictures that I've shown, you see like that there are these little valleys and ridges where cells localize. But this is because our particles are rugged. You can do the same with smoother particles, things that have been modified to have a smoother structure, or hydrogels or beads, and you don't see the same kinds of valleys. You see, like perhaps a more homogeneous distribution of cells, and I think that could be interesting to pursue.
Any other question? Okay, Jenna, sorry.
Thanks so much for your talk. Yeah, I was wondering too, like, you're not seeing films developing, like you don't think there's a diffusion limitation in those clusters?
Not in the cells that we're looking at. So because we have this very high detachment rate, a lot when we look under the microscope, it seems to be a monolayer of cells that forms, if ever. These cells detach super rapidly. So the things that I'm showing you, like the localization of cells, that's really rare to catch. We have other strains that are more sticky, so that form, like higher coverage of the particles, but even then we have never seen more than like maybe 2 layers of cells. So it's really, yeah, there are other studies. Knut Drescher, who's in Seoul now, I think they have this study on chitin particles where they see actually that the fusion within the biofilm is what limits growth, the fusion of oxygen and other gases. In our case, we don't think that's really happening.
And were you able, sorry to follow up, were you able to observe the detachment like with under the microscope? I'm sorry if I missed this. Like, I'm curious about your centrifuging method. Like, were you able to verify that you're not knocking them off?
That's a good question. So what we can measure is, a proxy of the single cell detachment rate, and then compare that to what we think that the population flux outwards is. And like these two numbers are related, but they're not exactly the same, right? So the faster microscopically a cell might detach might create a higher population flux of like detaching cells, but the timescales are not the same. So that's not something we're able to verify. What we've tried with the centrifugation method is I'm really centrifuging is at like 500 RPM on a small centrifuge, it's not even 1G. So it's really slow speeds, and we think that does not disrupt the localization of the cells to the particles. And in particular, because we're able to measure this exponential accumulation of the biomass on the particles using the same method, we think that, the method is not really disturbing the localization of cells. Yonesh.
Yeah, hey Ghita, thanks a lot. Very nice talk. Yeah, I particularly like how you just try to dissect everything, crossing out terms as you go. It's very cool. Maybe I missed it because my connection was not very stable, but you're proposing this trade-off between the replication rate, right, and the detachment rate. So are you thinking of this as having a feedback on the replication side or on the detachment side? Or is it sort of one and the same for you?
I think, let's see. I think if you think about what the cell is doing, the regulation is probably controlled by the nutrient concentration, which controls how fast the cell can replicate and grow, right? So the flux of glycnac into the cell is control its replication rate. So I'm thinking about the regulation really happening at the level of the replication. So this might not be true because detachment can also be potentially regulated through like the expression of pili that are retractable, and so what I'm thinking is, given a certain strain, it has a specific detachment rate. Perhaps it can regulate its detachment rate, but the thing that changes in response to the nutrient concentration is how fast it can double. And so I'm thinking that KD is fixed and RS responds to KD in this way.
I see.
Is that
And just very,
helpful?
yeah, it's very helpful. And just very briefly, can you mention how you were regulating the uptake of gluconate by the planktonic cells to measure the, what is it, KE?
Yeah. So the uptake of gluconate by the planktonic cells is actually quite low because of the fusion. It's not present. Experimentally, what we do is we add chloroform, chloramphenicol, something that prevents growth and replication. And so what we're thinking really we're doing is that we're preventing the uptake of the surface attached cells. The thing with the enzymes is that you can't separate the enzymes on the surface of the particle, right? So you have to assess both at the same time. And so that's, we think, the uptake that we're preventing, but of course, like it has the added possibility of preventing the uptake by the planktonic cells as well. But we think they're starving anyways, they're not seeing nutrients. So most of the uptake in the natural system would be happening on the surface of the particles. And so we can prevent these cells by taking up nutrients and then just look at how these nutrients accumulate in the media.
Okay. Marco, do you have a question?
Yes, I'm starting to realize that maybe I'm a bit confused about the whole stuff. I need to read the paper, I guess. But do I understand correctly that the idea where you might have a collapse on the population on the particles in the model, because I guess you have explored that only in the model, right? Is that is equivalent to you trying to essentially sub a culture at with a dilution factor which is higher than what the cells can, the maximum reproduction rate of the cell. Because if I understand correctly, what you have is that you have a situation where the cells detach very rapidly compared to how fast they grow onto the particles. And so this is similar to I have a batch culture and I try to dilute it, and I dilute it at a rate which is too high compared to what the cells can follow.
Yeah. I think that's a good intuition. So yes, you have a very high detachment rate where you need to, whatever, bacterial or biological effects you're going to express. So for example, you need to express chitinases to degrade the chitin, and you have to do that faster than your attaches away from part of it. That's what whether you can go attach rate you have to fight against, which is equivalent, I guess, if you're in a chemostat to a dilution rate. Chitinases be able to.
The follow-up question then, wouldn't then one expect that the cells should be able to regulate the detachment rate in order to avoid collapse in the population in the way that the model would predict?
Yeah, but we don't think that's possible, right? So you can say it's a bit more of a complicated model to think about, right? The simplest model is to say the cells are stuck with a certain detachment rate that's dictated by their degradation strategy. And so that's what they have to fight against. It is possible that they regulate the detachment rate, though in the literature, I've seen more biological ways of regulating attachment than regulating detachment. So you can, decide to become more sticky, make more TEP, have more pili. That's a way of regulating how much you attach. But we're thinking that for some reason you're detaching at a fixed rate. And that's what you're fighting against. If you introduce the regulation, then, there's no game to play.
Right.
You can be both very good at colonizing when you need to and good at detaching when you need to. And it's not clear that that's, that's a that cells can navigate. Yeah.
Thanks.
Go ahead.
You're muted.
You're muted.
Yes. Thanks for the talk. I have some sort naive question. do you have did you have like some sort of flow in your experiment, like with the culture? How does that affect this whole experiment? I'm not sure I followed exactly, but I'm just curious.
Yeah, so our experiments are in these shaking flasks, so there is some shaking that's happening, which might mean that, you know, in certain regimes, convection might be higher than some of these diffusive forces that we're thinking about. That's true on the one hand. On the other hand, we do think, we did like a few estimations, Jonas did help with this at some point, thinking about, you know, whether this really completely destroys the microscopic structure around the particles. And we think that that's not the case, that at least, you know, the localization of nutrients around the surface of the particles and of cells around the surface of the particles is conserved. We're a pretty low tech lab. I think, you know, one way of doing this is to put your particles in a microfluidic device and then subject them to controlled flow. And people have done this in Roman Stalker's lab in particular. And so like you can see different dynamics there in response to the flow. Yeah, we're being very crude about some of these estimates, I guess.
Any further questions? If not, let's thank Ghita for a fantastic talk. And we'll see each other here online in June. And for those of you who come to Australia, we'll see you in Australia.
Awesome. Thank you for
Bye.
having me. Bye-bye.
It was a pleasure. Thank you. Bye.